Circulatory System Part 3 ICC Class 10 Topics and Revision. WBCs. What WBCs are? The other cellular element present in the blood. What are the features of WBC? WBC has nucleus. Per cubic millimeter of blood, 4,000 to 8,000 WBCs we can see. It is amoeboid in nature. Then what is the shape of WBC? That is amoeboid. It produces pseudopodia during diapetesis. What is diapetesis? Diapetesis means squeezing out of WBC through the walls of the capillaries to the tissue fluid. Squeezing out of WBCs through the walls of capillaries, very important, through the walls of the capillaries to the tissue fluid is called diapetesis. While writing the definition, these terms you have to use, then you will get full mark. You don't miss the word walls, then capillaries, walls of the capillaries. These terms you have to use. Types of white blood cells. The technical term is leukocytes. There are mainly two types of white blood cells you can see. First one, granular WBC, that you can see here. These are granular WBC. Then the next one is granular WBC. These three are granular WBC. First, we can discuss about granular WBC. The cytoplasm contains granules. Nucleus constricted into lobes. Then here you can see in cytoplasm of granular WBC has granules. That's why the name is granular WBC. If there is cytoplasm contains granules, then that type of WBC we call it as granular WBC. Then you can see in this picture the nucleus is constricted into lobes. Then based on the lobes, these granular WBCs are classified into three. What are they? They are neutrophils, eosinophils and basophils. First we can discuss about neutrophils. What are the features of neutrophils? Here you can see nucleus with 3 to 4 lobes. Yes, with 3 to 4 lobes. Then it has granular cytoplasm. That also one of the important feature. It's a common feature for all the granular WBC. Then what is the function of neutrophil? That is phagocytosis. That means it engulfs bacteria. Then it produced in bone marrow. Then it's stained with neutral dyes. Very easy to remember neutrophil because it's stained with neutral dyes. Or one point, neutrophils means it's stained with neutral dyes. The next is type of granular WBC, that is eosinophil. What are the features of eosinophil? Nucleus with 2 lobes. In this picture you can see is nucleus with 2 lobes, granular cytoplasm, then function is phagocytosis. In all the WBCs, the common function is phagocytosis. Then in some WBCs have other functions, especially in eosinophils, it secretes antitoxins associated with allergy. The other features of eosinophil, that is produced in bone marrow, stained with acid dyes. Then what are the features of basophil? This is another third granular WBC, that is basophil. Nucleus large, indistinctly lobed. Nucleus is very large and we don't know how many lobes are it, right? In this picture you can see this, it is indistinct. Yes, nucleus is indistinct, large nucleus. Then another feature that has granular cytoplasm. Granules you can see in the cytoplasm. Function. It releases chemicals called histamine for inflammation. Then any inflammation, which chemical will produce? That is histamine. That is the feature of basophil. Then which dilate blood vessels. It will dilate blood vessels. Then it produced in bone marrow. Then it's stained with basic dyes. The basic dyes, because of that the name came basophils. Then you can remember easily neutrophils, that is neutral dye. Basophils, basic dye. The remaining one more is there, that is acid dye. That is, the name is eosinophil. Eosinophil stained with acid dyes, but basophil stained with basic dyes. And all are produced in bone marrow. Then these are the common features. Yes, granular, all the three has granular cytoplasm. But based on the lobes of nucleus, it is classified, mainly classified into three. Granular WBC. Mainly there are two features are there. Cytoplasm without granules, then a single large nucleus. Here in this picture you can see there is no granules present in the cytoplasm of this WBC. Then two types of agranular WBCs you can see. First one lymphocyte, another one monocyte. What are the main features of lymphocytes? Lymphocytes are the smallest of WBC. It has single large nucleus. You can see here lymphocyte has single large nucleus. What is the function of lymphocyte? It produces antibodies. This is also one of the important feature. It produces antibodies produced in bone marrow and lymph glands. Lymphocytes are synthesized in bone marrow and lymph glands. Then the another agranular WBC, that is monocyte. You can see that in this picture. Yes, that is kidney shaped nucleus you can see here, right? Yes, monocytes have kidney shaped nucleus. Then there is no granules, right? Cytoplasm has no granules. Then at the site of infection, it will transform into macrophages. Then what is the function? It ingests germs. That means it will engulf bacteria. That process we call it as phagocytosis. You already learned this term from circulatory system part 1, right? Then where it will produce? It will produce in bone marrow. Yes, then synthesis happens in bone marrow. Monocytes produced in bone marrow. Let us discuss about the functions of leukocytes. The main function is body defense. We can go through some points. Phagocytosis. This is one of the main function of all WBC. This you can mainly neutrophils is responsible for phagocytosis. What is phagocytosis? This is a process in which most of the WBC engulf particles like solid substances, especially bacteria. Then engulfing solid substances or bacteria, we call it as phagocytosis. An abnormal increase in WBC up to about 50,000 or more per cubic millimeter indicate some infections in the body. If any germs or bacteria enter in our body, the WBC count will increase about 50,000. Then through blood checkup, we can find out that all of you know that WBC, the normal WBC count, that is 4,000 to 8,000. The next point, inflammation in monocytes and neutrophils. These two are responsible for this. Inflammation occurs due to the localized inversion of germs or the reaction of tissues due to injury. You know that here you can see in this picture, any injury happens or any harmful things enter in our body. Here is these WBCs will squeeze out through the walls of the capillary. Yes, you can see that in this picture. This is a capillary wall. This through the walls of the capillary, it will squeeze out into the particular area. All of you know that that particular term, squeezing out of bacteria, that we call it as diapedesis. Through diapedesis, yes, this will migrate into the particular area. It migrates through the walls of blood vessels by diapedesis and fight against disease-causing germs. You can see this one clearly, right? The next point, formation of antibodies. The main function of lymphocyte, that is formation of antibodies. Here, yes, these antibodies kill or neutralizes the germs or poison. Then all of you know that you all got second dose vaccination. Then now it is familiar, right? The vaccines stimulate the formation of antibodies. All of you know that through vaccination, yes, in our body, antibodies are produced. WBCs will produce antibodies. Then antibodies persist long after the disease has been overcome. Then we will get the immunity power while we are getting vaccination. Origin and life of WBC. It is produced in bone marrow. Then the average life is about for 2 weeks. Then 125 billion neutrophils are produced every day, as its lifespan is few hours because of its lifespan is few hours. In every day, 125 billion neutrophils are produced. The old and worn out WBCs are destroyed in the spleen, liver, and bone marrow. Then these are the two terms, leukemia and leukopenia. Abnormal increase in WBCs, that we call it as leukemia. It's a blood cancer, right? Then here, this WBC, the number of WBCs are increasing. Then that phenomenon, we call it as leukemia. It's a blood cancer. Then if it is abnormal increase in WBC, that causes leukemia. Abnormal decrease in WBCs, the another is disease, that is leukopenia. Here you can see the destruction of WBCs. Blood platelets, the technical term is thrombocyte. This is the third cellular element present in blood. The other two you already learned, RBC and WBC. Let us discuss about the features of blood platelets. It's minute oval or round structures. It's a non-nucleated structure. There is no nucleus in platelets. It's very minute and oval in shape. Then 2 lakh to 4 lakh platelets you can see per cubic millimeter of blood. Platelets are derived from megakaryocytes in the red bone marrow. These megakaryocytes are in the red bone marrow. Then these platelets are derived from megakaryocytes. The lifespan is 3 to 5 days. It destroyed in the spleen. Important in clotting of blood. These blood platelets are very important in clotting of blood. Then what is the main function of platelets? The function of platelets, that is, it initiates the process of clotting of blood. While writing examination, at the time of examination, you have to write the term initiates. Otherwise, you will not get mark. This platelets initiates the process of clotting of blood. Revision, exam-based questions and answers. Some questions based on these topics we are discussing. Write the functions of the following. First one, WBC. What is the main function of WBC? Phagocytosis and yes, production of antibodies. The next one, platelets. What is the main function? It initiates blood clotting. Initiates the process of blood clotting. Then you have to write the term initiates. The next one, lymphocytes. What is the function? The main function is production of antibodies. Then the next one, neutrophils. Of course, that the main function is phagocytosis. Basophils. What is the function of basophil? Is again phagocytosis you can write. Then production of histamine. Then define the terms. The next one, define the terms. Diapedesis. What is diapedesis? Diapedesis is the process of squeezing out of WBC through the walls of capillaries into the localized area or in the infected area. That is called it as diapedesis. You have to remember squeezing out, that term you have to use. Then through the walls of capillaries, very important. These two terms you have to use, otherwise you will not get mark. First you have to remember, yes, through the walls of capillaries, then squeezing out of WBC. Then the next one, phagocytosis. Phagocytosis means what? WBC is engulfing the bacteria or substances. That process we call it as phagocytosis. Engulfing, that term you have to use. Who will engulf? WBCs. That also you have to mention. Then leukemia. It is leukemia, abnormal increase in WBC. What is leukopenia? Abnormal decrease in WBC. Name the following. 2 granular WBC, eosinophil and basophil, or neutrophil also you can write, any two you write. 2 granular WBC, eosinophil and basophil. The next one, 2 agranular WBC. that is lymphocyte and monocyte. Abnormal increase in WBC. Abnormal increase in WBC. The term they are asking, leukemia. That is the term. The next one, abnormal decrease in WBC. Leukopenia, the cell from which platelets are derived. Megakaryocyte, the smallest WBC. Which is the smallest WBC? Lymphocyte. The WBC with a kidney-shaped nucleus. That is monocyte. The WBC produces antibodies. Antibodies, that is lymphocyte. The WBC produces antitoxins. Eosinophil. WBC produces histamines. That is basophil. Thank you, children. Have a good day. Learn circulatory system part 1 and 2. Score good marks. Thank you.